Welcome, everyone. Thank you for attending this webinar on container closure integrity testing method development. My name is Marjolein Althoff, and I will be hosting today's webinar. I am joined by our panelist member, Josine Wilmer. She is study manager at Lighthouse and will be available during this webinar to answer all your questions. And I'm also joined by our presenter, Derek Duncan, who I will formally introduce in a few moments. Since the increasing attention from regulators and the industry to CCI testing, we decided to organize this webinar on CCI testing method development. With this webinar, we hope to provide you with a guide for CCI testing method development using Headspace gas analysis, and we will give you some practical tips using real data illustrations and case study examples. During the webinar, you can post questions by using the Q&A window on the right-hand side of your screen. Please do not use the chat function. And during and also at the end of the presentation, we will try and answer as many of your questions as we can. Then I would now like to formally introduce to you our speaker, Dr. Derek Duncan. Dr. Duncan began his career at the Dutch Institute of Atomic and Molecular Physics in Amsterdam. He then moved into industry holding product and application development positions. And currently at Lighthouse since 2003, Dr. Duncan is responsible for developing applications for process monitoring and finished product inspection. With that, I encourage Derek you to start your presentation, and I hope everyone will enjoy this webinar. Thank you.
Thank you, Marjolein, and welcome to everybody to this webinar. Looking over the registration list, I think we have people calling in from the U.S., from Europe, as well as Asia. So welcome, everybody, and good morning, good afternoon, and good evening. As Marjolein said, we will present today on container closure integrity testing method development, and specifically method development using laser-based headspace analysis. So I'd like to go through this material in the following framework. So the agenda for the webinar will be as follows. I'll start with a few slides to give a little bit of context to CCI testing and CCI method development, giving both a regulatory and an industry context. We'll then go into how to develop Headspace gas ingress methods for CCIT, and then we'll end and hopefully spend most of our time on some concrete case studies, which should give you hopefully some clear insight into how this can apply to your own work and your own CCI projects. And as time permits, we'll collect some questions for a Q&A session at the end. So first of all, a little bit about the regulatory context, and I've listed two regulatory documents here. The first one being USP 1207. This chapter is on package integrity evaluation and was implemented in August of 2016. If you have not read this chapter yet, I would strongly recommend it. It's an easy to read chapter and also very thorough. The old 1207 chapter was about 2 pages long, and this new one has now been divided into 3 subsections for a total of 50 plus pages, and it gives a lot of helpful and concrete information about CCI testing approaches and technologies. The second regulatory document listed here has to do with the EU Annex 1 revision. And the Annex 1 actually governs our sterile manufacturing guidelines for the EU. It is now in revision. A draft version has been publicized, and I just mention it here because there are some things said about CCI in this document, and not only about CCI testing in manufacturing, but actually throughout the product life cycle. And I was just part of a workshop discussion, which also included some inspectors that were involved in the revision of Annex 1, and there's a general consensus that this will have, could have significant impact on best practices for CCI testing. But I think to summarize, these two things and the way that the industry has been moving in the past few years, to summarize it, I think the regulators are paying more attention to the proper design of CCI studies, validation of CCI test methods, and also the generation of statistical data, which doesn't happen too often actually at the moment. We could have a whole webinar on USP 1207. In fact, we've done that in the past. If you're interested in watching that, I believe that we have copies of that webinar posted on our resource page on our website. But if I would summarize some of the three main points from this chapter, I would perhaps choose the following. There is clearly a preference for what the chapter calls deterministic CCI test methods, and these are mostly analytical, physical test methods. And there's a preference for these over the current probabilistic methods, and the chapter names the blue dye and the microbial ingress test as examples of these. The chapter also recommends performing CCI testing during the entire product life cycle as part of a coherent container closure strategy. So from development to scale up to manufacturing, and then during shelf life for a particular product, there should be a database of CCI data which helps you to manage and control the risk of CCI. And finally, it eliminates the requirement to compare these new deterministic methods to microbial ingress testing. Science-based data is sufficient to validate the method. And I think the comment on this would be that robust analytical methods that can be quantitatively validated are really needed to meet these new guidances that are found in this particular chapter. I just wanted to say something about this last bullet point as well, because I was recently in a discussion with an industry representative who mentioned that just a couple months ago they were inspected, and this inspector asked them for, they were using actually some of these new deterministic analytical methods for CCI testing, and they asked for the correlation to microbial ingress testing, which disappointed him a little bit, seeing that the chapter actually recommends that you don't need to do this. And in fact, it's a little bit contradictory with this particular guidance, which was released by the FDA in 2008, where they actually suggest using a CCI integrity test in lieu of sterility testing as a component of the stability protocol for sterile products. And what they are essentially saying here is if you can prove over the shelf life that your container closure is good, and you do that with a physical validated CCI test method, then that is sufficient to actually guarantee or to demonstrate the maintenance of sterility. And doing a physical container closure test method is actually straightforward, more easily validated, and in fact for this purpose is better than a sterility test. Now, historically, leak defects have been linked to a product sterility risk, which is why I think you see, as happened to this particular company, you see people wanting to link container closure to a microbial ingress test. However, more recently, more and more products have a requirement to maintain headspace gas content or vacuum, for example, for product stability and product preparation, and maintaining the headspace gas content is actually leads to more strict requirements for CCI. And you can ask yourself, what is a critical leak for my product? Obviously, for all sterile product, the risk of microbial ingress applies across the board. You have to keep your product sterile. For other product that may react with reactive gases such as oxygen or water vapor, moisture, the loss of critical headspace gases can lead to a loss of stability and product efficacy. And for the patient, of course, this is just as much a risk as microbial contamination. And really, when you think about it, a container that does not allow passage of gas molecules will also not allow the passage of microorganisms. So if you can develop a CCI test method which shows that your container is gas tight from, or at least is free of defects that allow for the free passage of gas, then this container will also be a microbial barrier. And you can see this also in these slides from a recent FDA presentation. Again, there is sometimes the idea that the microbial ingress test is the gold standard for determining the closure integrity of a container, of a sterile product container. However, you see in these slides, it says in the second slide here, there is no gold standard CCI test. The testing method selected should be appropriate for both the product and the final packaging configuration. And if you can read the smaller print in this third slide, a test method is validated if it has been proven through scientifically accepted studies to be capable of detecting a breach in container closure, system integrity. The test method should demonstrate the ability to detect a container breach that is worst case or critical. So again, there is a requirement to generate science-based CCI data to validate a test method. And it could be that one test method will be more applicable for a certain product than another. So there is no gold standard CCI test method. So let's look at current industry best practices. So from our perspective and in our interaction with our customers, very often blue dye and microbial ingress testing are the only CCI tests done during the product life cycle. But we see that the landscape actually in the industry is currently changing. And if you really want to follow USP 1207 and actually also the current draft EU Annex 1 revision, these current CCIT practices need to evolve to prioritize the use of robust analytical methods. And these more robust analytical methods will allow you to straightforwardly validate your test method quantitatively, and will also allow you to collect statistical data. So not just on tens of samples, but on hundreds of samples and maybe even thousands of samples, which the traditional methods would be very difficult to do. Of course, the question, what changes will be required by the regulator? So USP 1207 is a chapter that is above 1,000, so it is not a required chapter, but is seen as recommendations. And I think that's why we have right now a little bit of confusion in the industry, and the detailed best practices for CCIT are not clear. So I think that's what also generates the interest in a webinar like this one, and that's also what we see when we speak with companies who are performing CCI testing. We are in the middle of an evolution, I think, of best practices, and hopefully this webinar will give you some ideas of how you can evolve your CCIT practices in your work. But if I would summarize everything together from what we see at the moment, I think that end users are expected to generate science-based data with arguments in the context of QRM, of risk management, to justify CCI test methods and practices. And you see that in the new USP 1207. You also see that in the language of the revised EU Annex 1. Okay, so let's start talking about Headspace gas ingress. And what I show here, these bullet points, is how gas can in general flow through a defect in and out of a pharmaceutical container. And so there are basically two regimes. The first one here described is effusion. So this is gas flow that is generated by a total pressure difference across the container defect. So the pressure outside the container is different from the pressure inside, and this creates a driving force which can drive gas molecules in or out of the container. The second regime is diffusion. And in diffusion, gas flow of a particular gas is generated by a partial pressure difference of that gas across the container defect. So, or just a natural random walk migration of gas molecules. So you can have a container which has a 1 atmosphere headspace. You can have a storage environment where it's 1 atmosphere outside. But if that container has a defect in it, you will see gas flowing in and out of that container. What's nice about gas flow is that you can use gas physics to predict how this gas flow will occur. And understanding this gas flow then enables the development of fairly sophisticated CCI test methods which are based on the measurement of gas ingress. And so that's what we will talk about in more detail today. If you are not familiar with laser-based headspace analysis, I'll just use this one slide to explain the technology. So Lighthouse introduced this technology into the pharmaceutical industry almost 20 years ago. And what you do is take laser that is generated by a laser diode and you shine that through the headspace of a pharmaceutical container. And the laser light matches the frequency of a specific target molecule, so that when you measure the amount of laser light absorbed on the other side, you can actually determine the content or the concentration of that gas inside the pharmaceutical container. And this measurement is non-destructive, so the product is not lost, the sample is not destroyed, and can be remeasured if desired. And it's also a fairly rapid measurement, so you're talking about measurement times on the order of a second. And so you see that it can be very useful for packaging, integrity studies, and CCI testing. And it's also a very easy technique to use from an operator point of view. Here are some of the Headspace platforms that we actually deliver. The initial development of these platforms was actually funded by the FDA, and with some of their funding, we were able to develop 2 main product lines. So you see here benchtop instruments where you manually insert a sample for analysis of the headspace gas, and we also have these laser sensors integrated into automated inspection machines. The top picture is a pulsar machine, which is very compact and flexible, and I just wanted to mention it in this presentation because I think it's a new concept for the industry. You can actually lease this machine on a short-term project basis. So if you want to generate statistical CCI data or perhaps inspect a few critical batches, you can do it with this platform quite easily. And then we also, together with our partner Bosch, deliver high-speed inspection machines with the Lighthouse laser technology inside. So how do you use these platforms to generate CCI data? Let's start talking about that now. And in this particular animation, we have taken as an example a modified headspace product. So that means you have a product that has a headspace other than atmospheric air. And examples of these are lyophilized product. So almost all lyophilized product is packaged under a partial pressure of nitrogen or sometimes at full vacuum. If you have an oxygen-sensitive liquid, and you see more and more biopharmaceuticals, because they're large, delicate protein-based molecules, they have some oxygen sensitivity, and so these liquid products will be filled under a nitrogen blanket. So they're purged with nitrogen in the headspace and have very little oxygen there. You assume that these containers are going to be stored out in air, in ambient air. And so what will happen if a defect causes a breach in the integrity? Well, you can see this in this animation. Air from the outside environment will ingress into the container, and the speed of that ingress could be very fast if you have an underpressure, like a freeze-dried product. it would go slower if you have equal pressures inside and outside the container. But you see here in this graph that you will measure a rise in oxygen levels and a rise in the overall headspace pressure. Both the benchtop platforms and the automated platforms can be used very straightforwardly to do CCI testing for this type of product. A second example, and here you have product that does not have a modified headspace. You in fact have product with air in the headspace. So if you were out in an air environment and you have a leak, then air from the outside comes in and replaces air from the inside. You actually don't measure a change in the headspace content because it remains air. So here what you can do is actually put the sample inside a chamber that can be purged or evacuated. So now the idea is you want to drive a headspace change, and it's actually a very similar approach to blue dye testing, where you put containers inside a bucket with dye, you seal it and you overpressure it, and you try and drive those dye molecules through a defect into the container. Now you're doing it with gas, and it's going to be much more sensitive and will also be non-destructive when compared to blue dye testing. So you define some vessel cycle, a conditioning time, and then you remove the container and see if you've been able to trigger a change in the headspace condition. So what will happen here, let's say you have nitrogen or CO2 inside the chamber, you can overpressure it. This gas from the chamber will then make its way through the defect into the container headspace. And what will you measure? If you're measuring oxygen, you will see the initial atmospheric oxygen levels decrease because of this cycle, and you will see levels of your tracer gas, and let's say you use CO2 as a tracer gas, you will see the levels of the tracer gas increase. If you decide to evacuate this chamber, you could also do that. You will then cause the initial 1 atmosphere headspace inside the vial to decrease, and you will see a decrease in pressure. And all of these things can be measured with headspace analysis, and you can design an analytical CCI test using these principles. So I wanted to show you an example of a CCI test vessel, CCIT vessel, for headspace gas ingress testing. This is something that we actually provide. So you can see that it's a CE and ASME certified vessel. You can overpressure it to multiples of atmospheres. It can be evacuated, and you see the various gas inlet ports on this chamber or this vessel, and that enables you to backfill the vessel with a chosen gas atmosphere and then seal it. And then after you've put the samples through a particular cycle in the vessel, you can then remove them, and you use a headspace analyzer then to perform gas ingress testing, looking for a determination of the container closure integrity. And again, as mentioned before, this particular approach is very similar to blue dye ingress testing, but now you are detecting diatomic gas molecules which get into a defective container, and you're measuring that with a quantitative analytical technique which in the end allows you to generate very robust science-based CCI data. Now, I wanted to spend a couple of slides also on positive controls. All of these are elements that you will use in your CCI test method development. You could also do a whole webinar on positive controls, and we've also done that in the past. And I would encourage you to perhaps, if you're interested, look for that webinar on our website. I'll just go over a few slides here, and I've also shown here a number of references if you want to read further about how these were actually applied in real studies and experiments. So This slide just shows a few examples of positive controls. You see on the very left a glass micropipette, and it has a very small inner diameter and then is fixed with epoxy in through the glass vial wall. On the upper right there, you see a metal disc that was sealed onto the rubber stopper, and this metal disc had, again, a needle, a micro hole or a needle in the center, and that will then simulate a defective or leaking container. On the bottom right there, you see a copper wire that was sandwiched between the vial neck and the rubber stopper. Again, that would be an example perhaps of a CCI defect, a realistic CCI defect due to a fiber or some product that gets between the rubber stopper and the vial neck. Here are some pictures of some other controls, positive controls for CCI defects. A glass pipette here, which is a tapered glass tube, and you see at the very tip you can have a micron-sized opening. These are very delicate, however, and you see here how the tip has been damaged and starts to enlarge. In our laboratories, we have used a few different types of positive controls. On the bottom left, you see a microcapillary, which has a 10-micron inner diameter, and you can simply insert that through the stopper and glue it. But the glass defect on the right seems to be one that a lot of the industry is starting to, I would say, perhaps standardize on or use a lot, and these are laser drilled holes in a glass vial. So there are suppliers who will take glass vials and will laser drilled certified size defects in the glass body. What's nice about these defects is you don't change the configuration of the container. You can stopper it and cap it just as always. You can even fill product in these positive controls. And you can see from the pictures of these holes that they're not nice, consistent round holes. They are often measured with a leak rate, and this leak rate is certified or correlated to a size. But you can see it's a little bit of a tortuous path into the vial. And so one thing that we have done to try and understand how sensitive a gas ingress method can be is we have done some studies with a very thin metal disc, which is also laser drilled. And you can see the pictures of the holes that have been laser drilled into these metal discs on the right, and you see that they are actually quite nice round holes with a very easily measurable diameter. And what we use these for is as positive controls, we can then very confidently say in our study we were able to detect an ideal hole of 5 microns in diameter, and that's the utility of these types of positive controls. So they have very consistent, well-defined physical dimensions. So I think when you're talking about CCI method development, you're going to want to decide what kind of positive controls you use, because you will want to try as best as you can to determine the sensitivity of your method and to also show that you are detecting known defective vials versus vials that are sealed well. Here's some example of Headspace data that was generated using these positive controls. So what we are plotting here is actually the rise in Headspace oxygen over time of a vial that has been filled with nitrogen and also has one of these laser drilled defects in the glass body. So defects that are 5, 10, and 15 microns in diameter. And you can see from the time scale on the x-axis that we are monitoring the Headspace oxygen rise in days. You actually start seeing changes in Headspace immediately, but in this particular case, you can see that it starts to reach atmospheric levels for the larger defects on a time scale of days. And what was nice about this is that you can model this experimental data with gas flow theory. So I mentioned this at the beginning, and the solid lines are actually the results of some model calculations that were used, which take into account the volume of the container, the size of the defect, and the initial headspace conditions. And so that becomes a very powerful tool when you're trying to define an optimum CCI test method, and we'll see an example of that later on. And one other thing, we actually did a lot of work in this area, and there was a recent publication at the end of last year. So if you're interested in seeing all the data that was generated, as well as the theoretical background behind the modeling, I would encourage you then to read that publication. Okay, before we get to the actual industry case studies, I wanted to show one example of an internal study that we performed, and we did this to demonstrate the development of a CCIT method based on carbon dioxide overpressure. So the study involved 2 positive control sample sets, and in the first one, we had positive controls with these ideal laser drilled 5-micron defects, and they were drilled into a 15R vial. A measurement of the headspace CO2 was made to verify that there's basically 0 levels of CO2 initially in the vial. And then these vials were placed into a CCIT vessel and overpressured with carbon dioxide. And after this overpressure, we then took the samples out and we measured the headspace CO2 levels with a headspace gas analyzer. A second control set we actually filled with 10 milliliters of distilled water so that the defect was under the water level. Again, an initial measurement verified 0 oxygen levels at the beginning, and then after these samples were put into a CCIT vessel and overpressured with CO2, we looked to see if the headspace showed traces of CO2. And you can see here the storage was for 30 minutes inside a vessel, overpressuring with an absolute pressure of almost 2.5 bar of CO2. So if you look at a schematic in pictures, so a visual representation of this, number 1, the positive controls. You can see here a vial with a laser drilled hole in it. For one set, we kept them as empty positive controls, and for another set, we filled with water. These were placed in a CCIT vessel, and they underwent an overpressure cycle with CO2. The samples were then taken out and analyzed with a headspace analyzer. So let's look at the results. So this graph shows how much CO2 was measured in each of the positive control samples. I should also say that we had negative controls as part of the study. I'm not plotting them here, but they showed 0 CO2, both initially and after the overpressure cycle. So you see T0 plots in blue, and you can't really see them because they're 0. And then the T1 CO2 levels, so after 30 minutes in the CCIT vessel, you can see that for the empty vials, they show almost total headspace gas exchange. In other words, just about all the initial air inside these vials were replaced with carbon dioxide, and this is a clear indication that these vials have a leak defect. If you look at the results of the vials that were filled so that the defect was under the liquid level, we are also in this study able to detect all of the positive controls that were filled, however, with less sensitivity, because now you're forcing the CO2 gas through the liquid up into the headspace. And we actually did further studies which then showed there were cases where defects below the liquid level are not detected. And I know that there have been publications showing that if you have a product, for example, that has it's a high density proteinaceous product, biopharmaceutical, that actually the large proteins can actually clog a hole. And so you have a defective container with an integrity breach, but because something blogs or clogs the hole, a CCI test method that depends on gas ingress or gas flow in and out of the container will not detect that because the cloggage or the blockage blocks the gas flow. But that's just an example of why robust CCI method development is critical. And with this approach, using Headspace to make these measurements of analytical measurements of gas into a container, you can generate some very robust data to show where the method works, where it perhaps doesn't work, and also to try and quantify the sensitivity of the method. So we'll move on to the industry case studies, and the data in these previous slides are an example of combining positive controls with a CCI test method, and that's laser Headspace plus some kind of sample preparation, which involves a CCIT vessel for characterizing a leaking container. And these studies can provide a basis for CCI test method development and have great utility for determining the barrier properties of a package. And these studies enable not only a science-based validation of your method, but they can be easily applied to a variety of CCI applications. You see a few listed here, a QC CCI test. You might need to qualify raised stopper height specs on your filling line, CCI troubleshooting in manufacturing, determining appropriate vial stopper combinations and packaging development. So across the whole product life cycle, you can gain a lot of value from a quantitative, robust CCI test method. And that's what we'll look into now. So for the second half of this webinar, we're going to focus on some real industry case studies, and I hope that what you see in these case studies will give you ideas of how you can apply this to your own CCI projects. So the first case study I call a QC CCI test method development. A QC lab wanted to be able to receive samples of a specific product. It was a nitrogen purged liquid product, and do a CCI determination in less than 30 minutes. So the first thing that happened was we performed some oxygen diffusion tests. And you remember that diffusion means there is no pressure differential between inside and outside the container. And again, we use these laser drilled vials as positive controls. And if you look at the results here, so you see the plot of the rise in oxygen levels, again, as a function of time for these different positive control defects. So remember, these vials are initially purged with nitrogen, so you start with very low, 0 to very low oxygen levels. But because of a defect, you will see air coming inside of these vials, and this happens over a time scale of hours. So if you look at the x-axis, you see that we're plotting this over a time scale of hours. And the conclusion here is that the natural oxygen diffusion rates through these defects is too slow for this particular project, because the QC lab needed to detect these defective vials within 30 minutes. So now we took a CCIT vessel and we designed a CCIT vessel cycle to create an effusive condition, and that means we're going to try and drive changes in the headspace. And if you look at the graph here in the title of the graph, you can see that the cycle that was chosen was to put the samples into the CCIT vessel, evacuate for 20 minutes, and then backfill with air for 10 minutes. And you can see here that the requirement to detect an ideal 5-micron defect in 30-minute works. We are plotting in this graph the change in headspace oxygen for the different positive control samples, and you can see that all of the 5-micron defects were detected quite easily using this cycle, CCIT vessel cycle, and a headspace oxygen measurement. Here's a second industry case study, and I call this one a filling line CCI qualification. So very often when filling lines are set up, you need to generate some CCI data to demonstrate or prove that your filling line is processing the packaging components appropriately and putting them together so that you achieve robust CCI. So what we did in this case is had a manufacturer produce empty stoppered and crimp vials with the process. So there were very specific vial stopper combinations. Those were run through the filling and capping process, but were not filled with product. They were capped and crimped under a headspace of air, so that's 20.9% oxygen. And then we performed a CCI study, and we included positive controls having a 10 micron microcapillary through the stopper. We also used the gas ingress model to design an appropriate CCIT vessel cycle. And you see this in this table. So 3 different types of vials, and we could calculate how much, what the oxygen level should be as a function of defect size for these different types of these different vial configurations. So you can see that even for submicron defects, we should detect submicron defects with the chosen cycle. So the Headspace gas flow model can be used to calculate the oxygen levels as a function of vial size, sample preparation cycle, and CCI defect size. So if you look at the data generated from this study, so here's some example data from the 2R vials. There were 2 different vial stopper combinations, and you can see that the initial 20.9% Headspace oxygen levels stayed constant for all of the sealed samples, but the positive controls that were included in these studies showed a significant drop in the oxygen levels and were easily identified as leaking vials compared to the sealed vials. And so it was very straightforward to generate this filling line CCI qualification data. And you can imagine if you are a CMO and you are trying or needing to show to your clients that you can properly process all of their different packaging components to produce good CCI, it's nice to have a straightforward and very efficient and robust way to generate this data. Staying on the filling line qualification scheme theme, most filling lines these days are outfitted with a raised stopper sensor, and in the revised Annex 1 draft that is currently out, they added language which specifically says that you need to qualify your raised stopper height specification. It seems like a lot of people just pick a number and don't really have the justification behind that number. And so we've supported actually a number of clients who used Headspace CCI tests with stoppered vials that have shims, some plastic shims between the rubber stopper and the vial flange to simulate a raised stopper of various heights. In this study, we also used these laser drilled positive controls to simulate CCI defects and would compare the behavior of the raised stopper vials to these known defective vials. On the right, you can see the setup of one of these studies. We had shims of various thicknesses. You can see that you can very easily use statistically relevant sample sets, in this case 30 at each raised stopper height. We also included different types of positive controls as well as negative controls. And here's a graph that basically summarizes the results of this study. So what we are plotting here is that for each raised stopper height, we are plotting the percentage of leakers. And we had defined a leaker to be something that is leaking faster than a 5 micron positive control. And what you see here is that the correlation between stopper height and loss of CCI is probabilistic and not deterministic. And the shape of this probability curve we've seen from other studies can depend on your specific vial stopper combination. So you can see here that you generate very robust science-based analytical data to understand at what stopper height you want to reject at to properly manage the risk of loss of CCI in your process. Here is a case study which includes the use of an automated inspection machine. And in this case, a manufacturer was producing a freeze-dried product which was specified to be sealed at 200 millibars of nitrogen, so a relatively deep vacuum. And they actually had vacuum as one of their QC tests, and they noticed a number of QC tests failing, so they made a decision to perform 100% inspection of some suspect batches. And of course, in this case, you have to use a non-destructive technique. And so they configured a Headspace inspection machine. We actually rented this machine to them. I mentioned that as one of the machine platforms that we have, and it was configured for 100% oxygen inspection to identify lyo product vials that had high oxygen content and reject them as leaking vials. So in this case, you don't have to develop a very sophisticated method, right? Because this product has a modified headspace, there's deep vacuum. As soon as you have a leak, air will very quickly enter the vial, and a simple headspace measurement is actually your test method because elevated oxygen levels will identify leaking vials. And so here you see the data that was collected by the inspection machine. So this is data from 6 consecutive batches, and they show highly variable oxygen levels. This product should have 0 oxygen in it. You can see this data set is almost 180,000 vials. It was roughly 30,000 vial batches. And if you look at this analytical data more closely, you can see that you have a number of vials that have what we would call permanent leaks. That means that the headspace was totally replaced with air from the outside environment. But you also have a lot of partial leaks with partially elevated oxygen levels, and these are actually containers that were leaking during the process. So in the lyo chamber, coming out of the lyo chamber, they were leaking, but they reached the capping and crimping machine, and the leaks were actually stopped by capping and crimping. So this analytical CCI data gives really deep insight to reveal that the lyo sealing process is actually the issue and is not very robust. Another comment here, all of these partial leaks, because they are currently sealed, they will not be detected by a classical container closure integrity test. So blue dye, if you perform blue dye on these vials, they would pass because they are sealed at the time that you measure them. However, a headspace measurement clearly shows that there are a lot of vials here that are not meeting the specified conditions of the product. Okay, so I'll end with one last concrete case study here, and this describes a conceptual situation where you have a complicated device. So when you talk about container closure, you can talk about vials, and you think that that's simple, but actually it's not so simple. If you start looking at syringes and now devices, then performing a good container closure test can become a challenge. So here we have an auto injector with no optical access to the cartridge, so there's no window. And you can imagine the following. To perform a CCI test on this auto injector, you could place it in one of these CCIT vessels and develop a cycle that will drive carbon dioxide through any defect into the cartridge. And what you would do then is take this auto injector, inject the contents of the cartridge into a vial, into a secondary vial, and then test the headspace for the presence of CO2. And if you see any carbon dioxide into this vial, which you've injected the contents of the cartridge into, then that would indicate that the cartridge inside the auto-injector has been leaking. So again, if we look at this from a visual perspective, you have this auto-injector. You can use a CCIT vessel to run it through a cycle which is trying to force carbon dioxide into the cartridge inside. You take the auto-injector and you take a vial and inject the contents of the auto-injector into the vial, and then that vial headspace is measured in a headspace analyzer to detect the possible presence of CO2, which would then indicate a leak in the auto-injector. So I wanted to present this as an example of a little bit more complicated test method development, but it also points out how powerful this approach can be if you couple a non-destructive headspace measurement with a CCIT vessel. You can create a lot of different test methods that are optimum for various kinds of containers. So we're coming to the end here, and I'd like to summarize perhaps the main points. At the beginning of the presentation, we talked about CCIT best practices, and it's our general impression from what we see from the regulators and speaking with the industry that there is a need to evolve current CCIT practices to include analytical methods which are able to produce robust science-based CCI data. We've talked about, in particular, laser-based headspace analysis. There are other methods I forgot to mention, and you can also read all about them in the 1207 chapter. But this particular method is a rapid, non-destructive analytical method that can be applied to a wide variety of package integrity applications. And when you couple this technique with the use of positive controls and a CCIT vessel, laser headspace can be used to define a wide variety of CCI test methods which are really useful and add value through the whole product life cycle, whether you're talking about package development using empty containers, QC testing with real product containers, in manufacturing for either sampling or 100% inspection. And when you talk about using this technique with a CCIT vessel, I think this approach is very similar to current blue dye testing, but you can imagine it's much more sensitive. You're using an analytical method to detect small amounts of gas ingressing into the container. And what's nice about it is it's relatively straightforward to validate in a quantitative manner. Thank you for your attention. If I look at my clock, I was actually able to end on time, so we have time for a few questions here. Before we get to the questions, I need to show a slide from my marketing colleagues. We do support our customers with all kinds of CCIT activities, method development, feasibility studies. I could really only cover the tip of the iceberg on a lot of these topics. We also hold workshops, and the next one is being held May 16th at a location in the U.S. in North Carolina. You can go to our website for the details, and later on this year we'll be holding one in Europe. It's also possible for us to come on site if you're interested to give workshops at your facility. Okay, thank you again for your attention, and I'm being handed a few questions here which I can answer before the webinar ends. One question that's come in is it possible to develop this methodology for plastic bottles? The general answer is yes. The question with plastic bottles, of course, is that you will also have permeation of gas. And we didn't go over this in detail in this webinar, but you can easily distinguish between permeation and an actual leak defect. And so it adds an extra complication. So with plastic bottles, you have to take this into account, but in principle, it's possible. You also see a lot of activity at the moment with plastic vials that have a coating as a permeation barrier. This technique is also a great way to do fundamental package properties experiments to actually determine how well these coatings have cut down on the permeation. So yes, in general, plastic containers are this method can be applied to. Another question: if you have a liquid biologics fill at atmosphere with no headspace gas, how would you detect a leak? So every method has its pros and cons, and in fact, the USP 1207 gives some guidance in how you might go about choosing a method. Obviously, if you're using Headspace, you need to have a Headspace for it to be applicable. In this particular case, there are other methods that might be useful for CCIT in this situation, and they would include the high voltage leak detection method. It might be possible that some variants of a vacuum or pressure decay test could be useful for this type of configuration, product configuration. Let me see another question. What is the regulatory requirement for frequency testing of CCIT? Should each batch be tested? That is a very interesting question, and at the moment, it is not defined as a per batch test either in the US and certainly not in the EU. And we have, again, I was part of a workshop a few weeks ago, and this exact question was discussed. And in the context of the revision of the EU Annex 1, and actually the regulators themselves could not agree on this topic. So there were some regulators that expect to see some CCI testing data from each batch, but there are other regulators who think it is not necessary as long as you demonstrate that your process is under control. In both of these cases, the regulators want to see CCI data, and not only CCI data, but they want to see statistical CCI data. So if you are trying to prove to a regulator or inspector that your process is under control and producing good CCI, then you need to have some statistical CCI data that demonstrates that. So that is a point that they're able to agree on. And we'll see if that language stays in the revised Annex 1, but that is the indication that we're getting from them. And if they enforce that, then I think that will actually be a significant impact on best practices, because with the standard methods that are currently used, blue dye testing, microbial ingress testing, it's very, very challenging to produce statistical CCI data that give you insight into your process. I have one last question here before we wrap up and are out of time. And the question is the following: we currently only use Headspace testing during 100% inspection. Do you know of anyone in the industry that has submitted with only Headspace testing data for the initial qualification of CCI rather than microbial or dye ingress? I have to say, personally, I don't know of a specific example that comes to mind. I'm guessing, I'm sure there are. I do know of a number of customers who have submitted for the 2008 guidance, which was about doing CCI tests in lieu of sterility testing for the stability protocol. I do know of clients who have submitted Headspace as the CCI test in lieu of sterility testing, and that was accepted with no comments. But for the initial qualification of CCI, I'm not sure. If you read the language in USP 1207, they say that with your initial CCI qualification, the chapter says that you need to demonstrate that your test method well, first of all, you have to determine what they call the MAL, the maximum allowable leakage limit, and then you have to show that your test method will detect leaks down to that MAL. And they are talking about micron, submicron like leaks, and microbial and dye ingress will not be able to do that. So if you really follow the letter of the law in USP 1207, then you need to use one of these new analytical methods or deterministic methods. And in the USP chapter, they mention helium leak test and Headspace as the two methods having the sensitivity capability of detecting submicron leaks. Well, I hope those answers help. Again, we would look forward to follow-up questions after this webinar, and we thank you again for joining us, and we hope to hear from you soon.